Practical, Agarose Gel Electrophoresis, Working, Principle, Operation and Practical Guide. Aim, to perform Agarose Gel Electrophoresis for the separation and visualization of DNA fragments based on their molecular size. Materials required. Samples, DNA samples, genomic DNA/plasmid DNA, DNA ladder, molecular weight marker. Reagents. Agarose powder, 1x buffer, triacetate EDTA, Ethidium bromide, or safer alternative like SYBR safe, loading dye, 6x, distilled water. Equipment. Gel electrophoresis unit, tank plus power supply, gel casting tray and comb, micropipettes and tips, microwave/hot plate, UV transilluminator/gel documentation system. Principle, Agarose Gel Electrophoresis is based on the movement of negatively charged DNA under an electric field. DNA has a negative charge due to phosphate groups, so it migrates towards the positive electrode, anode. Key concepts. Separation is based on molecular size, smaller DNA fragments move faster and farther, larger fragments move slower. Agarose forms a porous gel matrix, acting as a sieve. Additional factors, Agarose concentration, voltage applied, buffer system, TE/TBE, DNA conformation. Working. DNA samples are loaded into wells of Agarose gel, electric current is applied. DNA migrates through gel pores. DNA is stained with dye, example Ethidium bromide, and bands are visualized under UV light. Procedure. Step 1, preparation of gel. Prepare 1x buffer from stock solution. Add Agarose, example 0.8 g in 100 ml buffer for 0.8% gel. Heat until Agarose dissolves completely. Cool to 55-60 degrees C. Add Ethidium bromide carefully. Pour into gel tray with comb and allow to solidify, 30 minutes. Step 2, setting the gel. Place gel in electrophoresis tank. Add buffer until gel is submerged. Remove comb carefully to create wells. Step 3, sample preparation. Mix DNA sample with loading dye, example 10L DNA plus 2L dye. Load samples into wells. Load DNA ladder in one well. Step 4, electrophoresis run. Connect electrodes, black equals cathode and red equals anode. Run at 100 to 120 V for 45 minutes. Stop when dye front reaches 3/4 of gel. Step 5, visualization. Place gel under UV transilluminator. Observe glowing DNA bands. Capture image if required. Results. DNA fragments appear as distinct bands under UV light. Smaller fragments travel farther than larger ones. DNA ladder helps determine fragment size. Example interpretation. Genomic DNA rightward arrow slow movement, large size, plasmid DNA rightward arrow faster movement, RNA contamination rightward arrow faint smeared bands. Conclusion. Agarose gel electrophoresis successfully separates DNA fragments based on their size. It is a reliable and widely used technique in molecular biology for DNA analysis, purity checking, fragment size estimation. Precautions. Handle Ethidium bromide carefully, mutagenic, always wear gloves, avoid air bubbles in gel, do not run gel at very high voltage, ensure correct electrode connection, use UV protection while viewing. Applications. DNA fingerprinting, PCR product analysis, genetic studies, cloning and sequencing.